Hi everyone, my name is Connor and you're very welcome to this Saturday session which is going to cover the Leaving Cert Biology topics of the scientific method and enzymes. Now just before we get into the topics, we're going to see where both of these units actually fit within the course structure. So if we have a look up here, we can see that the scientific method falls within Unit 1 and enzymes falls within Unit 2. Now we're just going to have a quick glance at the amount of topics that's within both of those units. So we can see that along with the scientific method, food and ecology are the only two other chapters or topics that fall within the Unit 1, whereas enzymes there is quite a bit more, but they're still vastly smaller than Unit 3. Now, why is this important for us? We'll have a look at that now. Now, as we move from the course structure to the exam layout, I'm going to talk about the Leaving Cert Biology paper prior to the COVID changes. There is word coming through that the 2023 class, so if you're sitting the Leaving Cert exam in June 2023, there is going to be changes for your exam as well. So this is still going to be relevant, but there will be additional choice then on top of that as well. We'll just wait for more word to come in on that. Now, we know that the scientific method falls within Unit 1, and 2 questions from Unit 1 will appear within the short questions. The scientific method is a very popular one to appear in there as a full question, and we'll be looking at an example of a question in a little while. Normally within the experiment questions, the scientific method would appear as a part A, so rather than an entire experiment question based on it, you might be asked something to do with the scientific method as the first part of the question. Normally it wouldn't be any longer than that. We know that enzymes then will appear in Unit 2, so again there's going to be 2 questions from Unit 2 in the short answer questions. Enzymes is a very popular one to appear there as well, and even more common it appears in the experiment questions, because we do have our 4 enzyme experiments which appear: the effect of temperature on enzyme activity, the effect of pH on enzyme activity, enzyme immobilization, and then the heat denaturation of enzymes as well. So very important to have our enzymes for the experiment questions. And again, we could be asked a little bit about the scientific method within the long question, never really would appear as a full question, and enzymes absolutely could appear there as a long question, has done so in the past. So just important to keep that in mind as we move through this topic, that they are really, really important topics and do pop up on the final paper quite often. Now we're going to get into the content, so we're going to start off with the scientific method. You would have seen this from the very beginning when you entered junior cycle science in first year, and you will be following these steps every single time that you complete an experiment within class. So if we have a look over here at the diagram on the right hand side, these are the steps that are involved within the scientific method. So we would make an observation, we would form a question, we would question why that is happening, we would form our hypothesis, we'd begin to conduct an experiment, which is our series of steps to test our hypothesis. We would form our conclusion based on all the results that we gathered, and then we would publish our results. Sometimes as well we could add in an extra results in here just before the conclusion, so I'll just write that in. So when you're carrying out your experiments, the layout that you might have followed is you've done your experiment, you've presented your results, you form your conclusion. This extra result here at the end is that you would publish your results to either be peer reviewed or for anybody across the world to read. So you could add in a second results there if you wanted to. But in essence, the scientific method, it's a process of investigation in which problems are identified and their suggested explanations are tested by carrying out experiments. So really important to have a definition for the scientific method, so important that we do know that one. Now this is just all the steps within the scientific method, just presented in a different way. So we would make our observation, we would form our hypothesis, which is our educated guess. You could also say it's a proposed explanation. And once you've formed your hypothesis or educated guess, you're going to test that hypothesis. And that there is going to be your experiment. We'll come to the principles of a good experiment in a little while, but we would have to make sure that it's fair, it's reliable, and that we have a control. Now, once you've carried out your experiment, you're going to collect and interpret your results, and you're going to form your conclusion. And once you've formed your conclusion, one of 2 things are going to happen. If the hypothesis is supported, this can lead to new knowledge. So as you're carrying out your Leaving Cert experiments, you will form your conclusion at the end. That's not actually going to lead to new knowledge because these experiments have been done countless times with students all across the world. But if it was a new experiment and it was done for the first time, it could lead to new knowledge. Now, if your hypothesis is not supported, you will have to go back up here. A new explanation will need to be found and tested. So we would reform a hypothesis and carry out the exact same steps again. So again, scientific method, what steps are involved? Well, we're going to make our observations, so we're going to notice something happening in the world and make our observation, formulate our hypothesis. We're going to design a controlled experiment. It has to be controlled. We have to have a control in there so we can compare our actual tested one to. We're going to collect and interpret our data. We're going to form our conclusion, compare the conclusion with existing knowledge, and we're going to report and publish the results. And possibly this could lead to the development of theories and principles, which we'll come to now in a couple of minutes. So we've already mentioned the word hypothesis there quite a bit. And the hypothesis is your educated guess or proposed explanation based on an observation. And the most important thing for a hypothesis is that it has to be testable. You have to be able to test it in a lab situation. So we have 4 examples of hypotheses here at the bottom. We have people exposed to high levels of ultraviolet light have a higher incidence of cancer than the norm. We have that goldfish make better pets than guinea pigs. We have it doesn't matter whether or not you skip class. And we have if you put people in a dark room, then they will be unable to tell when an infrared light turns on. So some of those are testable, some of those are not testable. We might be able to make them testable to a certain extent by changing the wording of them, but you could have a quick read through those now and decide which ones for yourself are testable. Now, as you read through them, you're probably looking there and thinking that the first one and the last one are testable, and I would agree absolutely with you, whereas the second one and the third one aren't testable in their current state. What do we mean that goldfish make better pets than guinea pigs? What are we actually measuring? It's too vague, it's too subjective at the minute, so we would have to set ourselves some parameters within there. What are we actually going to measure to ensure that goldfish are better pets? We'd have to reword it, so maybe you could say that goldfish are cheaper to keep as pets than guinea pigs. We have to set parameters that we can measure. Exact same down there for it doesn't matter whether or not you skip class. So at the minute, we can't actually test that. What are we testing? We have to set the parameters for ourselves. We have to find the variables. And then once we have those, we might be able to test it to some degree, but they're very, very difficult to actually to test on. Now, a couple of these words we've mentioned already, and it is important to know them because they do pop up as short answer questions in the section A, and sometimes again at the start of the experiment questions. And it's just the idea of what an experiment is, what is actually data, and what is a replicate. So the experiment itself is just a series of steps that's carried out to test your hypothesis. So a series of steps carried out to test our hypothesis. The data, that's just measurements, observations, or information that's gathered from the experiment. And we could go one step further than that. I'm going to do this up here on the right hand side. We could split our data into quantitative and qualitative data. So our quantitative data is data involving numbers. So that might be the rate of enzyme activity. It could be the rate of photosynthesis. So if you were doing the LADA experiment, you'd be counting the number of bubbles. If you're doing your ecology study, you might be looking at the percentage cover. So you're counting the amount of plants that are in a particular area. Whereas the qualitative data that's data involving words. And one of the experiments where you would have gathered qualitative data is also in your habitat study, where you were recording whether or not a particular organism was present. That was just yes, it's present, or no, it's not present. You weren't actually counting, you were just identifying them with a key. The replicate then is really, really important. So an experiment, it has to be repeatable, and you are going to carry out repeats in the experiment so that you can ensure fairness and accuracy. So the replicate here, that's just a repeat of an experiment or procedure. So you're going to carry out multiple of these as you carry out your experiments. Normally, as you were doing them, you would have done each experiment probably 3 times. So a couple more definitions here just to get through. We've our theory, which we mentioned earlier on. We said that knowledge or carrying out our experiments could lead to new knowledge, and eventually that could develop into a theory, a principle, or a law. So we'll come to that as well. We have our variables and we have our controls down there at the bottom. Now, a theory up here at the top, that's a hypothesis that has been supported by many different experiments. Okay, so one famous example which you might have come across in junior cycle already is the Big Bang theory. We have the theory of evolution and so on. If that theory has shown to be valid over a long period of time, then we can say that that becomes either a principle or a law. So within genetics, you would have already looked there at the law of segregation. The law of independent assortment would be another one there as well. If you're already studying chemistry, you might have come across Boyle's law. So if that theory is shown to be valid over a long period of time, it'll become a principle or law. A variable then is a factor that can change during an experiment. So we can actually break that down into a couple of different types of variables. So we have our dependent variables. We have our independent variable. And we have our fixed variable. So the independent variable is what you change. The dependent variable is what you measure. And the fixed variables are what stays the same. So we can break variable down into three types. Dependent, what are you actually measuring? The independent, what are you changing? And then the fixed variable, what's staying the same. Normally, we would only have one changeable variable in each experiment, just change one thing. That's why when we talk about the enzyme experiments later on, we change temperature in one and we change the pH in another. We don't change both in the same experiment. We do two separate experiments, and then the dependent variable is what you measure. Everything else stays the same. The control then, that's what you're going to use to provide a standard or comparison against in which the experiment can be judged. So a lot of the experiments you're carrying out, you might have used water as a control, but it's what you're using to compare your actual experiment to. So we mentioned on the last slide there how the experiment was a series of steps that we carry out to test our hypothesis. And when we're carrying out these experiments, it's really important to ensure that they're fair. So a couple of different ways that we can do this. Really important that we have a large sample size. So if you're carrying out a qualitative studies, maybe a social science studies, that you have a large number of participants who are responding, or if you're actually doing an experiment that you repeat it a number of times. So in class, you probably would have done it 3 times, but again, in the real world, we'd want to do that a lot more. Make sure that we carry out random selection to prevent bias. So that was really important when you're carrying out your habitat studies. When you were placing your quadrat, you would have thrown a pencil over your shoulder and then placed the quadrat down over there just to make sure that everything was random. And that's the same for any experiment where we can carry out random selection. And we have to make sure that the experiment is repeatable. No point having just a once-off fluke of an experiment. We have to be able to repeat that time and time and time again. Now, interestingly here, this is one that might have come up in the news over the last couple of years when I was coming to vaccines, the idea of double-blind testing. So double-blind testing is where neither the tester or the person being tested on knows who's receiving the treatment. So if we are talking about vaccines, the vaccine is obviously going to be the treatment. The placebo is one that's not going to have any effect. So I'll just write that word down for us, the placebo. So that's what it's being used to test against. It's not actually going to do anything for us. Where the double-blind testing, so neither the tester or the person being tested know who's receiving the treatment, just to make sure we're removing any bias there, and also to make sure that the experiment is as accurate and as fair as possible. Now, we mentioned earlier on about how when you're carrying out your experiments, you gather results and then you form your conclusion. But then once we formed our conclusion, ideally we're going to publish our results again. So the second time we're going to mention results. So the results of experiments should always be written down and reported on. Why is this? Well, this is so that they can be read, interpreted, and reviewed by other people. So peer review is really, really important. And anyone who goes on to third level, who might study science, or indeed any subject at third level, any paper that you might write is going to be peer reviewed just to check that everything is okay and that it's accurate, reliable, and fair. So publishing experimental procedures and results such as in journals such as Nature and Science, they'd be the big ones, means that new ideas are available to all scientists and it can grow everybody's scientific understanding. So really important in this day and age that we're sharing everything that we do just to make sure that knowledge across the world is ever being increased. Now, the scientific method does have some limitations. So some of these limitations, it's limited by issues such as the extent of our basic human knowledge, our ability to interpret the results, that can only go so far. The scientific method is also confined to what can be observed with the 5 senses. So an example of that is what existed prior to the Big Bang and the known universe is literally outside of our realm of science to investigate. We just cannot do that. Changes in nature as well. So one example of that will be antibiotics. When they were first invented, they were originally thought to kill all bacteria and could well have. but there are some bacteria that now are resistant to those. And one way that that would arise is if you don't finish your course of antibiotics, the bacteria then that are left over in your body, even though you might be feeling better because you're near the end of your course, you just haven't finished it, they can start to mutate and then eventually become resistant to those antibiotics. So really important that we finish out our courses. And it can also be limited by accidental discoveries. So one of the most famous examples we have there is around the discovery of penicillin. So we know that the person who discovered penicillin was Alexander Fleming. And at the time of that, he was actually carrying out work in St. Mary's Hospital over in London. He was doing a lot of work on bacteria over there. More specifically, he was carrying out experiments on the Staphylococcus bacteria, which you might have heard before. He'd actually gone away on holidays, and when he came back, he noticed that there was a mold that was growing around the bacteria, but it seemed to actually be preventing the bacteria from growing. So there was a ring around it where the bacteria couldn't actually get into, or didn't seem to actually be growing into. So he started to carry out some tests on that particular mold and realized that it was inhibiting the bacteria. It was forming a chemical to protect against the bacteria, and that led to the discovery of penicillin. So saved countless lives, but he didn't actually mean to discover it. It was completely accidental. He'd left his experiment unattended before he went on holidays, came back and discovered penicillin. Now, we can also talk about ethics when we talk about experimentation and the scientific method. So when we mention the word ethics, it refers to whether an issue or conduct is right or wrong. So some of the things that might have popped up in the past is the use of animals in experiments ethical? Is the use of cloning or the carrying out of cloning on animals, is that ethical? Medical issues such as contraception and the use of stem cells raise ethical issues and is quite a large debate in the scientific method, more so the use of stem cells. And then the idea of organ transplants from animals to humans. So readily we do carry out organ transplants from humans to humans, but there is a debate there, would it be ethical to start to do that from animals to humans as well? So here's an example of an exam question which has come up in the past. So this one is from 2019. One of the short answer questions was question 2, and we can see it relates directly to the information that we just covered. A full question on the scientific method, so 20 marks. First question there, what is meant by the term hypothesis? So words such as educated guess would have been 100% perfect to write down for that. And we also mentioned earlier on, we did say it was a proposed explanation as well. So there are 2 possible answers that would have got you full marks. Explain what is meant by double-blind testing. So this is where neither the tester or the subject knows who's receiving the treatment. Down here, explain the necessity for random selection. Well, one possible reason would be to reduce bias. That's one answer, and I'll throw in a second one there as well. It would also increase the reliability of the experiment as well. Give 2 other features of good experimental design. So we can't mention anything that's already been mentioned. So what could we say here? Well, the fact that we could have a control. We could also say that we only have one independent variable. So that's the variable that you change, so you're only changing one thing. We'll have a look at a couple more there as well. So it has to be repeatable. You have to be able to repeat the experiment. It can't be a once-off fluke of an experiment. And we have to have replicates in there as well. Okay, so loads of possible answers there. We just needed the 2. Just make sure you don't mention the idea of double-blind testing or random selection there, because they've been mentioned in the previous answers and they weren't actually accepted that year. Where are the results of scientific research usually first published? So you could say a scientific journal. So that's just a general term. If you wanted to be more specific, you could name them. So 2 that we mentioned there was Nature or Science. They were the 2 scientific journals we had mentioned. And why is it important that scientists publish the results of their research? Well, so that they can be peer reviewed, they can be checked. And we throw in a second answer there as well. Also increases the knowledge of people. So really important that we do that. So we can see one full question there in the scientific method, covering everything that we've seen up to this point. Now we're going to move on and we're going to start to have a look at the idea of enzymes. So this was the topic that appears from Unit 2. Normally comes up as an experiment question, but can do so as a short answer question and a long answer question as well, which we'll see towards the end. So what do we mean when we talk about the idea of metabolism? So metabolism is the sum of all chemical reactions that take place within an organism. So we know that there's a countless number of chemical reactions taking place within our bodies, and metabolism is a collective term for all of those reactions. Some of these reactions involve growth and reproduction. Now we're going to just quickly talk about sources of energy before we get into discussion of what the enzymes actually do. So sources of energy, we know that solar energy or the sun is the primary source of energy for all of us. And cellular energy, that's energy that's stored in the bonds of biomolecules. So think of your foods, your carbohydrates and your lipids. So that's your cellular energy. And it's this energy that's going to be passed along the food chain. So if you remember from ecology and the food chain, a lot of energy is lost as heat as organisms feed on each other, but the energy that is passed on is the cellular energy. Now, enzymes, this is the big term for today. We want to know exactly what they are. And when we talk about enzymes, we're saying that they're catalysts. So a catalyst is an enzyme or a substance, I should say, catalyst is a substance that speeds up a chemical reaction without being used up itself in the reaction. Now, enzymes, their biological nature, they are proteins. So you can refer to enzymes as two different things. We can say that enzymes are biological catalysts, or we can say that they're protein catalysts. So biological catalysts, they're substances that speed up chemical reactions without being used up itself, or you can say they're protein catalysts because they are protein by nature. So two options there. Quite simply, you don't need to know all of that. They're either just protein catalysts or biological catalysts. Now, there's a couple of key words here to do with enzymes, and we're going to have a look at the enzyme action down here at the bottom. So the substrate is the molecule that the enzyme is going to act on. So that's your substrate. The enzyme is going to act on that. The product is what's going to be formed. So they're any molecules that are produced at the end. And the active site, that's the part of the enzyme that joins to the substrate. That's what's going to carry out all of the work and produce the products. Now we're going to have a quick look at the diagram down here just to get an idea of enzyme action, and then we'll go through it in text in the next couple of slides. But we can see here at the start, we have our enzyme down here. This portion of the enzyme here is called the active site, and that's where the substrate, so the molecule that the enzyme acts on, is going to join to, and eventually we want to form our products. Now I just want you to note at this point here at the shape of the active site on the enzyme and the shape here at the side of the substrate. We can see that they're roughly similar, okay? It looks like it's going to fit in there perfectly. And that's really, really important because the enzyme's active site is going to be complementary to its substrate. So I'll write that word down, complementary. And what we mean by that is the enzyme will act on one type of substrate. So there is a reason that they are similar shapes. The enzyme is only going to act on one that does have a similar shape, also known as specificity. Or we can say that the enzymes are specific. Now down here we can see that the substrate has joined to the enzyme, so the active site, it's been locked in there. And because the enzyme has been joined to the substrate, that's going to form what we call the enzyme. Apologies about the spelling. Enzyme substrate complex. So when they're joined together, we get our enzyme substrate complex. Now the enzyme is going to start to act on the substrate in here, and eventually we've formed our products. In this case, the substrate was large and we've ended up with two smaller products. Exact same thing could happen the opposite way around. We could have two substrates coming in here. They could be joined together to form a bigger product. So you can go from a big molecule to a small one, or smaller molecules could be joined up to bigger ones. And we'll get to a discussion on that in a couple of minutes as well. But the idea here, the enzyme, the active site is going to be complementary to the substrate. It's going to be specific to it. It's only going to act on one type of substrate. When they join, we're going to get our enzyme substrate complex. The enzyme is going to start to act on the substrate and our products will be formed. Now our enzymes, they're made up of amino acids. We know that because enzymes are protein in nature. Amino acids are the building blocks of proteins, and they're folded into a 3D shape. So we've already mentioned this already, but an enzyme will only work with one particular substrate. And normally, if you're ever reading scientific articles or if you're reading through experiments, you're normally able to spot which of the chemicals might be an enzyme because they will end in ASE for the most part. So some examples which you might have come across already. If not, you might see them before the end of 6th year. We have lipase, we have amylase, and we have maltase. So amylase is the enzyme that acts in your mouth. So amylase acts on starch to produce maltose. Now we've seen this already, but we're just going to talk through it with text beside the diagram here. So the idea of enzyme action, or the induced fit theory. So the active site is going to have an approximate shape of the substrate. It most likely isn't going to be 100% exact. And it doesn't have to be 100% exact because the active site can slightly adjust or change its shape. So the active site is going to change slightly when the substrate joins to make an exact fit. And we can see that in the diagram down here. We can see that roughly these portions of the substrate will fit into the enzyme, not 100% exact, but the active site here can change so that it does fit perfectly. And once the reaction is finished, the active site can change again and return to its normal shape, so it will look exactly like this again. So again, what are we saying here? We're saying that it's complementary. We form our enzyme substrate complex because the active site changes shape. Now, we've already mentioned there a couple of minutes ago about how when the enzyme is acting on the substrates, we can go from a larger molecule to smaller molecules, or vice versa, we can go from smaller molecules to a larger one. And that gives rise to two types of chemical reactions. We can have catabolic reactions, and the second one we're going to look at in a minute is the anabolic reactions. So the catabolic reactions, or the catabolic enzymes in particular, these catabolic enzymes will break a larger molecule into smaller molecules. And an example of that was the amylase we mentioned a couple of minutes ago. So it's produced in the salivary glands in your mouth. It's going to convert starch or break the starch down into maltose sugars, which are smaller molecules. And we can see that happening over here. We have our enzyme. We can see that the active site is actually nearly a perfect shape there. It's going to join to the substrate, form the enzyme substrate complex, and it's going to break down the starch, which is a complex molecule, into smaller ones, which is maltose. So that's a catabolic enzyme. It's a catabolic reaction. We're going from bigger molecules to smaller molecules. The opposite of that then is the anabolic enzymes, or anabolic reactions. So this is where we can convert two or more simpler molecules into a more complex molecule. So an example of this, if you've studied DNA already, you'll come across DNA ligase. If not, you will see it before the end of 6th year. And this is used in joining two pieces of DNA together. So we want to form our new strands of DNA. We can see here we have our two strands, and the DNA ligase is responsible for joining the two of those, going from two smaller molecules to a bigger molecule, which in this case is going to be our DNA strand. DNA polymerase then, that's found in plants and animals, and it can form and repair DNA as well. So two examples there of anabolic enzymes. Now, there are a couple of factors that will affect enzyme activity, and these are part of the experiments which you will conduct as part of your studies of enzyme as well. And the two experiments that you will look at are temperature and pH. So the effect of temperature on enzyme activity and the effect of pH on enzyme activity. So if we have a look over here on the right hand side, we can see along the y-axis, that's the rate of enzyme activity, and down here on the x-axis we have temperature. So there isn't actually any numbers here given on temperature. We won't worry about that. We'll mention that over in the paragraph here. But in general, for the first while, as temperature increases, so too does the rate of enzyme activity. So as the temperature is increasing, the rate of enzyme activity is increasing, and eventually we reach a point where the rate of enzyme activity is at its optimum. It's doing the best that it possibly can. Once we go above that temperature, and in humans, those enzymes will work best at 37 degrees, so body temperature. Once we're below or above that temperature, the rate of enzyme activity is a little bit lower. But what's really important here is if we go too far above the optimum temperature, the enzyme can become denatured and it won't work anymore. So the active site will be rendered unusable. The enzyme just will not work. So it's optimum around body temperature, about 37 degrees. If you go too far above, it'll become denatured. pH again, so the optimum pH in humans is between a pH of 6 and 8. Reason for that being that a neutral pH is around 7. Lower and higher pH values can lead to denaturing as well, but some enzymes will be adapted to work at the extreme values. So some enzymes out there will work well at pH of 2, others will work well at a pH of 9, maybe even higher. So we can see here roughly between 6 and 8, that's the optimum pH there. Enzyme activity will be at its highest. It'll go down below and above that. But if we look at the diagram down here, there's 3 examples of enzymes here. They don't end in the ASE, which we mentioned earlier on, but we've pepsin, amylase and trypsin. So amylase is the one found in the mouth. So we can see there it's about a pH of 6, which makes sense because we want a neutral one in our body, which is around 7. Pepsin, though, down here, it works in the stomach, which we know is a highly acidic environment because of the hydrochloric acid that's there. And that actually works best at around a pH of 2. And that would make perfect sense because of the environment that it's in. Catalase would work well at about a pH of 9. And trypsin, we can see here, if we were to bring that down to the x-axis, somewhere between 8 and 9, that's going to be the optimum pH for trypsin. So in general, optimum pH is between 6 and 8, but there are some enzymes that are adapted for higher or lower pHs. So the previous two slides, we talked about the effect of pH and temperature on the rate of enzyme activity. That's 2 of the experiments which you're going to do as part of your study of enzymes. The third experiment you're going to do is to look at the heat denaturation of enzymes. And then the fourth one is the production of immobilized enzymes. So what are immobilized enzymes? If you've already done the experiment, you might remember these beads, which we'll talk through in a couple of minutes. If you haven't, that's an experiment which you will be doing. But in general, these immobilized enzymes are enzymes that are attached or fixed to each other or to an inert material. So where are they used? Well, they're normally used within bioprocessing, and that's the use of enzyme-controlled reactions to make a product. All of these reactions normally take place within a bioreactor, which is just a vessel in which the living cell is used to make a product. And traditionally, this would have been used to grow, to make cheese, yogurt, and bread, but now can be used to produce antibiotics and drugs. So if we have a look at the experiment over here on the right-hand side, we can see we have our enzyme and alginate. What you probably would have used in this experiment was yeast. So yeast normally contains 2 different types of enzymes. That would have been mixed in with the alginate gel. And then this mixture would have been dropped down into calcium chloride. And once this mixture hit the calcium chloride, the calcium alginate beads will have the entrapped enzymes inside them. So those beads are what we can use then to test for the presence of the enzyme. So where or how can we actually immobilize these enzymes? So there's a couple of different ways you can do it. So you can entrap them, which is what you will do as part of your experiment for the immobilized enzymes. So this is where the enzyme is trapped in insoluble beads or microspheres, such as calcium alginate beads. So that's entrapment. That's the one that you need to know, and that's the one that you will carry out. There are 2 other ways as well. So you can immobilize enzymes through cross-linking. So this is where enzyme molecules are covalently bonded to each other to create a matrix consisting of almost only enzymes. So we're essentially joining the enzymes to enzymes. And then we can also do it in a carrier-bound method. So this is where the enzyme is bound covalently to an insoluble support, such as a silica gel. So it cannot be dissolved. So that's an example of carrier bond. So what are the benefits of immobilized enzymes? Well, one of them is that they can be reused again and again and again, so it's cost efficient. When these enzymes are added to the substrate, we're going to form our products. If they were free enzymes, we would have to separate the enzyme at the end, but because these are solid, we can separate them quite easily. So that saves some money there as well, so it's cost efficient, and it's also cheaper than using the free enzymes, which we just mentioned. Now, where are immobilized enzymes used day to day? So we know that glucose is often used as a sweetener in foods, so example in soft drinks. Fructose is a much sweeter but very expensive sugar. So if we wanted to get really sweet drinks, ideally we might use fructose. An immobilized enzyme though, called glucose isomerase, so again ending in ASE there, just to let us know that it is an enzyme, glucose isomerase, is used to convert glucose to fructose. So we don't actually have to purchase the fructose to put in our soft drinks, or the companies wouldn't have to do that. They can actually just use this enzyme to convert the glucose into the fructose, saving a lot of money. So enzymes can also be immobilized on paper or plastic strips and used to detect or diagnose specific substances. So an example of this, a clinistick, which is the name of the company that actually produces them, and then it's also the name of the stick. So a clinistick contains an enzyme that changes colour when it reacts with a specific chemical. So in this way, they can be used to test for substances such as pregnancy hormones in urine. So the idea of a pregnancy test all boils back to just enzymes. So we're going to have a look at 2 exam questions then based on what we've covered in enzymes so far. So this one came up as a part B of a long question, and we can see there that it says the single most important property of enzymes is their ability to increase the rates of reactions occurring in living organisms. So what is an enzyme? So we have 2 possible answers here for this one. So you can either say a protein catalyst, because they're protein in nature, or you can say it's a biological catalyst. You don't have to get into an explanation of catalysts there, but we do know that they can increase the rate of reactions without being used up themselves. So part 2, which type of molecules are enzymes? Or biomolecules are enzymes. We've actually pretty much answered that up there already. So we know that they're proteins. Part 3, name 2 factors that affect enzyme action. This was the 2 experiments which we talked about. So we have pH and we also have temperature there as well. What is meant by the term specificity in relation to enzyme action? We can say there that the enzymes act only on one type of substrate. Okay, they're specific, act on one type of substrate only. Part 5, name the part of an enzyme responsible for a specificity. We know that's the active site because that's where the shape can change. And part 6, give a detailed account of how enzymes worked. So when this came up, it was actually worth 9 marks, so we need to know 3 different points. If you're a little bit unsure, remember biology is done in multiples of 3. So we had 3, 6, 9, 12, 15, 18. So there's 9 marks left over. We need 3 points, so we'll put down as many as we can. So we could mention the previous point which we already made about how enzymes act on one type of substrate. We'd say that the active site changes shape. The enzyme substrate complex is formed. So we've mentioned the changing of the shape. We will mention then that the products are formed. And then we throw in an extra point there about how the active site returns to original shape. I'm not going to get it fitted in there, but the active site returns to the original shape. So we had 5 points there, only 3 were needed, but again, always write down as much as you can remember. So answer the following questions from your knowledge of enzymes. Again, I'm not going to do all of these because I've done some of them already, but explain enzyme specificity with reference to the active site. So the enzyme will only act on one type of substrate, and you could mention there about the active site having a rough shape, which will be specific or complementary to the substrate. What happens to the rate of enzyme activity when they're placed in a medium outside of their optimum pH? So we will say that it reduces, so enzyme activity will reduce, and the reason for that is the active site shape changes or can be denatured. Part 3 there. Name 2 substances used in the school laboratory to immobilize enzymes or yeast cells. So we'll say there we had our sodium alginate. That's what the yeast was mixed with, and then that was added into our calcium chloride. And then 2 advantages of immobilized enzymes: they can be reused, and they're also much cheaper. So that's all of the content for today, but before we leave it, I just want to show you around the ExamRevision website. Now, once you log into the ExamRevision website and go to Leaving Cert Biology, this is what you're going to see. So we can see there we have video tutorials, we've quizzes, presentations, we've our exam builder, and we've our resource pack. So if you wanted to find the content from today, all you would have to do if you wanted to go back over the videos is tap on video tutorials. We know that enzymes are in Unit 2. It's within cell metabolism, and we can see there we have videos on enzymes and their structure, photosynthesis and respiration. If we go down further, there's a quiz there on enzymes, so you can click into that and test your knowledge of enzymes. And if we go back and go right down, there's presentations or PowerPoints there, but there's also a full set of notes for you to access. So everything that we went through today, there's H1 notes, really succinct and defined notes for you to go through to make sure that you're getting the grade that you want. So everything that you might need is within there. And it's not just for the topics that we did today, it's for absolutely everything. So it doesn't matter which unit we go into. If we go into the Unit 3, which is the organism, we go into responses to stimuli, we can see that everything you need to know is right there. So if you want to find out a little bit more information about that, all you have to do, go to the ExamRevision website, go right to the bottom. There's a little bit there about the plans and the pricing. You can have a flick through that. There is an option for monthly subscriptions as well, so you don't have to sign up for it all in one go. You can pay as you go and then leave it whenever you want. So everything you need to know for Leaving Cert Biology and all of your other subjects as well can be found on the website. So some of the ones we have Ag Science, we've Business, Chemistry, Economics, English, French, Geography, History, Irish, Maths, both for higher level and ordinary level, and we also have music and physics as well. And if you do have any questions, feel free to get in touch, but I hope you enjoyed the session, and don't forget they're held every Saturday on the YouTube channel, so make sure that you have them saved, you're checking in, and watch it back whenever you get a chance. So I've been Connor, thanks a million for joining in, and I'll see you again soon.